cocaine-abstinence mediated behaviors. So, as mentioned earlier, 1.4 million people met the diagnostic criteria for cocaine use disorder in 2021, and cocaine use disorder, or CUD, is chronically relapsing, making treating CUD very difficult. And indeed, there are no FDA-approved pharmacological interventions to treat CUD. Because of this, there's a need for further understanding of the biological mechanisms underlying cocaine relapse. So glucagon-like peptide 1, or GLP-1, is a satiety hormone and endogenous modulator of the mesolimbic reward system. It plays a key role in regulating both homeostatic and hedonic feeding, as well as energy balance and gastric emptying. Specifically, food intake causes an increase in endogenous GLP-1 signaling, and activation of GLP-1 receptors causes a decrease in food intake. So it's kind of like a feedback mechanism. And because of these properties, GLP-1 analogs are FDA approved to treat diabetes and obesity. More recently, however, the emerging body of evidence suggests that they may also play a role in drug-mediated behaviors. So our lab in particular has started looking at the role of these GLP-1 receptors in cocaine reinstatement in rats, which is an animal model of cocaine relapse. So how do we model cocaine relapse in rats? Well, it consists of these 3 different phases that kind of mimic the human cycle of drug addiction. So first, the animals self-administer, the self-administration phase, the animals are allowed to lever press for intravenous infusions of cocaine, and this models compulsive drug taking. And then during extinction, the cocaine syringe is replaced with saline, and cocaine taking is extinguished, and we note that as less than 15% of drug paired or active lever presses. And then last is our reinstatement, which models relapse. So the animals are given an IP injection of cocaine directly before being placed back into the operant box and being re-exposed to drug pair cues that were paired during the self-administration phase. And when this happens, their engagement with the active lever, the drug paired level, will increase, effectively modeling cocaine relapse. So we use this model, and we pre-treated the animals with an IP injection of Exendin-4, which is another GLP-1 analog, and we saw a really nice dose-response curve in the ability of Exendin-4 to decrease engagement with the active lever, effectively decreasing cocaine reinstatement. And while these were exciting, the exact neural mechanisms and cell types responsible for this effect remain widely unknown. So the central amygdala is a complex brain nuclei previously implicated in a lot of withdrawal phenotypes, specifically the ability of cocaine, drug paired cues, and stress to reinstate cocaine seeking. And of interest to our lab, it also receives GLP-1 producing projections from the NTS and expresses GLP-1Rs at high levels. So first I wanted to see if Exendin-4 was even making it into the amygdala, so I injected them with a fluorescently labeled version of Exendin-4 and found that it did indeed cross the blood barrier and localized to neurons in the central amygdala. Next, to study the role of GLP-1Rs in cocaine reinstatement, we infused Exendin-4 directly into the CEA 10 minutes prior to that priming cocaine injection and the re-exposure to cues, and found that it dose-dependently attenuated the ability of cocaine and drug-paired cues to reinstate cocaine seeking. However, we were a little, cautious with these findings. GLP-1 is FDA approved to treat obesity and it decreases food intake. So we also looked at feeding behaviors to make sure that they weren't adversely affected, including pica response, which is another feeding behavior quantified by kaolin intake. And basically it's a chalky white substance that the animals will eat if they're feeling nauseous. So it's a way we can look at nausea and malaise in these animals as well. And we found specifically that the low dose that previously attenuated cocaine reinstatement also did not have any adverse effects on feeding. Next, I used RNA-scope to figure out what these neurons were, the cell types. So I found that about 65% of GLP-1R-expressing cells in the central medial region of the central amygdala also express GAD mRNA transcripts. And about virtually 100%, which is pretty much all the samples we looked at, in the central lateral and central capsular regions. So majority of GLP-1R neurons in the CEA are GABAergic. And we use this information to explore how exendin-4 and cocaine alters the activity of these cells. So I infused a pre-dependent GCAM virus into the CEA of GAD-free rats, and this allowed me to get a proxy of population-level neuron activity. So here's a control trace where the animals received, an IV injection of saline 10 minutes before an intravenous infusion of saline. And we see no impact on the calcium transient under this condition. However, when the animals received an IV injection of saline and then an IV injection of cocaine, we see this increase in area under the curve and mean Z-score, indicating that cocaine is activating these GABA neurons in the CEA. And most surprisingly, when we systemically treated them with exendin-4 prior to the IV infusion, rather than saline, we actually, at these behaviorally relevant doses we've seen before, we actually see that it turns neuron activity back to that of saline. So it reverses the cocaine-induced increases in GABA neuron activity. And then we also looked at, the last condition we looked at was exendin-4 followed by a saline infusion, and found an increase in mean score from baseline, basically suggesting that exendin-4 has some differential effects on these neurons in the presence or absence of cocaine. I've also done a series of tracing studies to look at where these GLP-1R GABA neurons project, and found that majority project to the nucleus accumbens. Specifically here in the yellow, you can see that 59% of cells that express GLP-1R and GAD transcripts were accumbens projecting. And I also identified some populations that project to the VMST and the VTA. So I wanted to answer the question of if the CEA to NAC GABA pathway was, activating that pathway was sufficient to attenuate cocaine reinstatement. So to tackle this question, we used a chemogenetic approach where we infused a pre-dependent dread virus into the CEA of GAD-free rats. And then the rats were allowed to self-administer on a normal schedule, and during the reinstatement test sessions, they received an intra-NAP infusion of either CRO or vehicle, and this allowed us to selectively activate the dreads that were expressed on CEA to NAP GABA terminals. And when we do this, when we activate this pathway prior to the reinstatement test session, we do get an attenuation of active levodopa, suggesting that this pathway is sufficient to attenuate cocaine reinstatement. And this effect is not seen in the M-cherry control animals. So in addition to cocaine reinstatement, we're also interested in looking at other cocaine abstinence mediated behaviors. Specifically, I've been interested in looking at the anxiogenic effects of cocaine withdrawal. So given that the CEA also plays a key role in anxiety phenotypes, we have started to pilot an experimental paradigm to explore cocaine abstinence induced anxiety using the elevated plus males. So the animals underwent cocaine self-administration, followed by 14 days of forced home cage abstinence, upon which they were then placed into the elevated plus males. These animals also had a yolked saline pair, so they received an equal number of infusions of saline, and so we used those to compare between the two. So after the 14 days, they got one treatment, and then after a break, they got the next treatment the next day. And so for my saline animals, we see that exendin-4, and this is a similar finding to what the other labs have seen looking at that EPM, is that it decreases time in the open arms and open arm entries in these saline drug-naive experienced animals, suggesting that it's anxiogenic. However, in the cocaine experienced animals, it actually seems to have the opposite effect. So the animals spend more time in the open arms and more open arm entries, which was really exciting and kind of surprising, these two differences here, because it kind of suggests that maybe changes in endogenous GLP-1 signaling could be the thing mediating this effect. So whatever changes are caused by cocaine taking and subsequent withdrawal are contributing to the presentation of anxiety during withdrawal. So to summarize, activation of GLP-1Rs in the CEA attenuates the reinstatement of cocaine seeking. Systemic exendin-4 suppresses cocaine-induced increases in GABA neuron activity. CeA GLP-1R-expressing GABA neurons project to the NAC, and chemogenetic activation of these neurons is sufficient to attenuate cocaine seeking. And systemic exendin-4 produces an anxiolytic effect during withdrawal in cocaine-experiencing animals. And taken all together, these findings not only further support the potential for GLP-1 receptors as molecular targets for novel drugs to treat CUD, but also kind of highlights a novel role for GLP-1 in mediating the anxiogenic effects of cocaine withdrawal. And yeah, so thank you to my lab, the Schmidt lab at UPenn, and then also PERF. So a lot of the undergraduate funding I got was really what started this project. And then lastly, the BBC conference. This is the first time I've ever presented in like a forum like this. So it was a really great experience for applying to grad school and everything. So yeah, thank you.
I actually have a quick question. Great job. So I was just curious why, for the elevated plus maze studies, you waited until 14 days.
Yeah, so I've looked at 7 days as well, and I don't have this data presented here just for time's sake, but we don't see that anxiogenic effect at 7 days. So it really seems to be a 14-day out, which kind of makes sense when you think about incubation of cocaine craving, right? Those cravings are longer, 60 days, 90 days out than they are 10 days out. So it seems to be, yeah, like it's a longer thing. But yeah, we found that, no change in anxiety behavior, and then also like adrenal gland weight per body weight was only increased at that 14-day time point. Yeah.
So I know these GLP-1 drugs that are approved for use in humans are approved for subcutaneous injections. Do you know if they're orally bioavailable? I guess I'm wondering if they are effective treatments, if there still might be some hesitance to prescribe an injection medication to at least this subset of people that are injection drug users.
Yeah, so that's like I guess in terms of, that's a little bit more relevant in terms of like maybe opioid use disorder, right, or other injections, but there are, or at least attempts to make like oral medications. I think there's one out there right now that's FDA approved, but I don't know if it is as effective in terms of like drug-mediated behaviors.
Thanks.